Do, do, do. Wait, my trusty sidekick is present. Mm, Uh-oh. See if I can get the audio. You're not going to do anything to the cat. Yeah, she's telling to go away. It's all right. She's not going to do anything to you.
So if you're in speaker view and you click on the three dots on someone's like little square with their initial in it at the top, and click hide non-video participants, it will get rid of everybody that is not currently using video.
Oh. Why do you want me to do that?
Oh, just for anyone who's like, oh, I'm seeing so-and-so instead of the professor. Like that will get rid of everybody except for you because you're the only one with your camera on.
Oh, okay. Is that not something that each person can do individually?
Yeah, I was just saying it out loud because it's too much to
okay.
type.
I got you. Yeah, I might need to do that if I need to do any drawing or anything like that, so I can see what I'm doing a little bit more easily. Or we could just watch the cat-dog standoff go on for as long as it does. Anyway, yeah, well, my dog would come out of it the worst, trust me. And her dumpster sister is even more vicious. She's actually drawn blood before. But, you know, dogs are pretty stupid, so they don't learn. Oh, right. So essentially, we're starting the new section today. You know, I'm going to leave the, I'm not going to hide video participants for now because it's pretty handy way of figuring out someone talking or writing something. Oh, yeah. Hey, you know, I actually read, I've watched Fight Club. Has anybody actually read the book Fight Club? It's really good. It's actually a lot better than the film, and the film was pretty awesome. Yeah, my opinion as well. Same for I Am Legend. Can't remember who that's by. But that was also a really, really cool book. And a good film too, but a really cool book. Anyway, let's see if I remember how to... Dum dum dum. Oh, hey, I'll tell you something funny. Just saw on the news this morning that Jair Bolsonaro, from the president of Brazil, has tested positive for COVID, which is pretty ironic, given how much is said that it isn't an important thing. Anyway, so essentially what we're going to do today is get started on our next section, which includes, actually, holy crap, there's going to be an exam next Thursday. Whew. This is going to be a pretty wild ride till the end of the semester, to be honest, peeps. It's basically a bit of kind of eukaryotic structure, chromosome structure, genome structure, things like that. And gene expression in huge detail, right? So we're going to be looking at gene expression as an overview. We're going to be looking at transcription in detail, and what is the stuff that goes along with that, such as RNA processing and stability, things like that. And then we will go through translation, right? And so These are, again, topics which have a lot of detail to them. They're very mechanistic. So, hey, cat, shut it, will you? And so you just, I don't know, it's just stuff you need to learn, basically. And part of the reason is there's so much else hangs on top of that, because essentially gene expression is what ties your genotype to your phenotype, right? In terms of what gets expressed when, in response to what happens if that doesn't work, you know, for whatever reason. Are really, it's kind of ties together, you know, really, really small biology and really big biology as well. And so that's why it's such an important topic to spend time on. And that's pretty much going to be the whole of the next section. So today, which is just going to be one lecture, we're going to talk about genome structure and transposons, which I think are pretty cool. I actually worked on them for a couple of years back in the day. Thursday is going to be transcription. Next Monday is going to be translation, and then it's going to be exam 3. Right. So try and keep on top of the material, because otherwise it kind of can be overwhelming. All right. So anybody got any questions about what's coming up? Let's see. How do I get the chat back up? Here we go. Let's stick that. So I'm just kind of getting used to how Zoom works in terms of where things go. All right, and again, no, don't do that. And again, don't forget exam next Thursday. It's a tough course to do over the summer because it's pretty relentless, right? There's just no let up in terms of homeworks, exams, assignments. Hopefully the lab stuff will be kind of not die down, but it will go back to the rhythm that we had before in terms of assignments. So that should clear up a little bit of time, hopefully, for you. Okay, so come on, there we go. This is actually a really pretty picture. I always think these are neat. This is what's called a karyotype. And so this is basically a picture of a genome. And so they, you can label different chromosomes with different antibodies, fluorophores, things like that. And they basically just take a picture. And this, here's a fun question, right? What stage of the cell cycle is this picture taken at? Think about what you're seeing and how many of everything there are. And if you look really carefully, some of them is a little easier to see than others. Look at 19, for example. Maybe the X, perhaps number 2. You got to look carefully at them. Wait, stop it. It's got to be prophase, yeah, of mitosis, right? So what are the, good job you put an R in there, Gabrielle. It would look really, really weird if you hadn't. What are the clues? There's 2 of them. What are the clues? We know it's not meiosis 2, right? Because we have an X and a Y, right? For example. So it can't be after reductional division has occurred. What's the first really obvious clue? It's actually so obvious that you might not get it. Is this what your chromosomes look like normally during stationary phase? No, because these chromosomes are super condensed. They're condensed for M phase, right? I don't actually, it's Yeah, I'd actually say that this is mitosis. It's not meiosis one, because these aren't linked up next to each other. Actually, Gabrielle, they're linked up. They're lined up by size. That's artificially done. That's done by like cutting out the picture of one and lining it up to the matching one. There's no way to actually make them line up like this in a cell. This is just an artifact. So the fact that they're super condensed is one clue. What's the second one? If you look really carefully at, say, the right-hand one of chromosome 2, can you see that ever so faint line down the middle? You can also see it in 19 on the right, maybe X as well. So that faint line basically tells you that each one of these consists of 2 sister chromatids, right? So that means that not only have these condensed down, but they've also replicated as well, right? So anyway, it's just neat. It's essentially, it's like a family portrait type picture. But it's quite cool to actually line all these chromosomes up by size and actually compare them, right? Because now you can see that they are not all the same, right? We have, yeah, they actually think it's very pretty. And actually there's an awful lot of art in science, right? It might not be kind of conventional stuff. And I'm sure Andy Warhol wouldn't have gone nuts over this, but but you can see, like, for example, a really cool comparison between X and Y, right? X is actually a fairly large chromosome. It's kind of equivalent to like the top 5 or 6 in terms of size. Y is tiny. And we'll actually see what that means in terms of gene numbers and stuff later. And they're also vary hugely in size, from 1, which is enormous, to 22, which is actually very small. And so this is kind of an artifact of evolution in terms of how these chromosomes were formed. And so typically speaking, and this is a very, very rough rule, chromosome count increases with organismal complexity, right? So flies and nematodes have, shit, how many do they have actually? They have 6. Flies have, I think they have 6 as well. No, they have 4. I don't actually know why flies have fewer than nematodes. Anyway, it's a rough rule, obviously. Chimpanzees have 20, I think, 21. And we have, oh yeah, thanks, Angela. And we have 23, obviously. So essentially that arose through a series of chromosomal duplications and mergers and splits. And actually there's quite a lot of dynamism there, if that's the right word, in terms of chromosomes over evolutionary history. In fact, in chimpanzees, potentially one of the reasons why they have fewer than we do is after our last common ancestor between us and chimpanzees, the chimpanzee branch actually had a fusion between 2 of their chromosomes. I can't remember which ones they are, but essentially made one much longer chromosome, right? So this isn't a static deal, right? It changes over time. I don't know what my dog is doing at my feet. However, all right, so that's a general rule, and it's a very rough one, okay? But when you look at genome size, totally goes out the window, right? You cannot make any general rule in terms of genome size and complexity. And this is a really great example. It's a really nice graphic, actually. And this is called, which we get to here, the C-value enigma or C-value paradox. C is the size of your genome, right? So ours is about 3 billion base pairs, which you can see here. And there are protists, right? Single-celled eukaryotic organisms, whatever Sarcodina are, right? Which are many times bigger than ours, our genome, right? And clearly we kind of are more complex than single-celled organisms, whichever way you shake it. And there's also a huge range, plants particularly, right? Animals tends to be a little bit more of a narrower range, although there's still huge variation. Protists are off the wall, and also plants are hugely varied. Part of that is because plants often undergo whole genome duplications. For example, wheat, don't know if you know this, but wheat is one of the eukaryotes with the largest genome size. I think it's 120 billion base pairs, something like that. It's absolutely crazy. And it's wheat, right? You know, we grow it, we grind it up, make, you know, bread or tortillas out of it. And the reason for that is that it's actually a hexaploid. This is cool. Writing a new word, hexaploid, if I can spell it. So it actually has 6 sets of chromosomes. We have 2 sets. We're diploid. And some organisms are quadruploid as well. I think there's something funky that happens in flies. Not 100% sure about that, but I remember seeing weird pictures of chromosomes lined up and stuff. But anyway, you cannot make any kind of comparison in terms of what something looks like and how big its genome is, right? And here's another nice example. This is a takifugu, so this is pufferfish. Don't know if everyone's seen the episode from The Simpsons, which is, I always forget to dig it up, where Homer eats pufferfish and gets paralyzed or something for a little while. I don't know what that is. It looks like a snake, but I don't think it is. It's some kind of reptile, right? So pretty similar in terms of what they look like, you know, apart from one's got fins and stuff. But a huge difference in genome size, right? They're both vertebrates. Yeah, legless lizard. I think it's got a name of some sort. It's not a skink because those have legs. These are really, really small ones. I should ask my daughter. She's nuts about reptiles. But anyway, and one of the reasons for this, and this is also another, I mean, you could get into like this whole field of kind of comparative genomics and evolution, right? You know, why we are the way we are at a big scale, is that very little of our genome actually encodes protein coding genes. I think, generally speaking, I'd have to check the numbers. I think it's about 3 to 5%, yeah, of our genome called protein coding genes, give or take. I'd have to double check to make sure of the exact number, if we actually know it, because it's kind of hard to figure out sometimes. Things like pseudogenes and stuff like that. And so main reason for that, right? And the reason between essentially, I don't know, how can we put it? So there's 2 different ways of thinking about this. One, you could compare 2 organisms which have essentially the same, have very different genome sizes, but look very similar, right? And so the easiest way to explain that is not in terms of protein coding genes, but in terms of how much other stuff they have there. So this used to be called junk DNA, right? And that's largely because we didn't understand, you know, what it does, and some of it doesn't do anything at all. But most of our genome and most organism genomes consist of not protein coding genes, right? Whatever that consists of, a whole bunch of different repetitive DNA, typically, transposons, all kinds of like evolutionary cruft that's accumulated over time. The exception to that tends to be organisms that are small and parasitic. Those tend to have much more compact genomes, generally speaking. So that's one way of thinking about it. The other way of thinking about it is to take 2 organisms which are very, very different and have approximately the same genome size, so, and same gene count. So I don't think actually, let's go gene count, might be a little bit easier. So C. elegans, my favorite model organism, is a nematode, right? So it, eats bacteria and rotten fruit and stuff. And humans have approximately the same number of genes, right? So both, oops. Give or take. And yet we're vertebrates, we have, I wouldn't say we have more complex behaviors. Maybe we do. Kind of depends on your perspective, I guess. But clearly we're far more complex at an organismal level than soil nematodes are. And yet we have the same number of genes. And so how would one explain that? Right. And so in that case, this really comes back to what we'll be studying later on in this section. It's not just what genes you have, and there are differences actually between C. elegans genome and ours in terms of genes. They have way more chemosensory genes than we do, for example. They have a much more, much better repertoire for sensing their environment than humans, similar to dogs in a sense. Where was I thinking that? Oh yeah, so same number of genes, but also how those genes are controlled is much more complex in humans than there is in worms. And there are many more copies of each type of gene. So for example, worms have one particular signaling pathway, right? They have one of each gene in it. Humans may have 4 or 5, right? Copies of that through duplication over time, essentially, gene duplication, right? So it's quite a complex deal, right? When you think about it in terms of, why are we the way we are, right? It's obviously there's the environmental aspect, which we're not going to talk about much here, but it's what genes do we have, right? What other stuff do we have in our genome, right? Which may not be protein coding genes, but may control those genes, right? So things like non-coding RNAs, for example, which are much better understood now than they were, say, 20 years ago. And also how complex is the control over those genes, right? How many bells and whistles and knobs and dials do humans have to control the expression of those genes versus simpler organisms, right? And those bells and whistles have largely arisen through natural selection over time, right? Over evolutionary time. So there's a lot going on essentially, and that's why you can't really look at an organism's gene count or its genome size and say this fits within this particular part of the tree of life in terms of complexity and evolution, right? It doesn't work that way. It's much, much more complex. And even when we look at individual chromosomes, right? It's not uniform. So, for example, here's Y. I'm not picking on the guys here, but Y is a very small chromosome, and it has very few genes. Or if you remember back to when we were talking about sex determination, it's largely genes that promote testes development and genes that are involved in sperm production. That's pretty much it. Right? How it got to that size, who knows, right? I mean, I don't know if it started bigger and then just lost bits that weren't necessary, which seems a little harsh. But then you compare that to, for example, that's got 6 genes per million base pairs. Then we have other ones. Here's chromosome 19, which is not much bigger, right? It's what, 70 versus 50 million bases. It has 1,700 genes versus 128, right? So, same gene density, it should have, what's that, like 200, give or take? It's got many times that. So it has 27 genes per million base pairs. Yeah. Well, I mean, it could just be that guys are more efficient, right? They just don't carry around a load of genetic baggage they don't need. Yeah, that I never win that argument, that's for sure. So, and this is also kind of a relic of evolution, and also these repetitive sequences and things called satellite DNA, right? So essentially throughout the genome, but some places more than others, there's whole stretches of DNA which are repeats, right? And so they could be just single repeats, or they could be thousands and thousands of times repeated. And part of that is those repeats actually cause problems sometimes with DNA replication. So, what is it? Yes, ALS, Lou Gehrig's disease, which is a neurodegenerative disease, which causes all kinds of problems. That is a trinucleotide repeat disease, right? So typically, at least, it's oops, gotta spell this right. And I think it's polyglutamine. So this is actually in a coding region, so it's not a perfect example. PolyQ. Q is the symbol for glutamine from memory. But essentially what happens with, so these repeats may have been caused by duplications. They could have been caused by transposons as well, which we'll get to in a little bit. But also they can be caused by the DNA polymerase stuttering. And so when DNA polymerase hits these repetitive sequences, sometimes it kind of produces more of them than it should do based on the template DNA. So essentially, if you've got a run of, say, 3 triplets that are all the same, instead of just copying those 3, it adds another one in, right? And that then gets incorporated into the next round of cell division. And so you get this expansion of these repeat or repetitive sequences. And in ALS, it's based on the number. I think it's something like 30 or 40 of these repeats is asymptomatic. And obviously, as time goes on, right, repeat number is going to increase through successive rounds of cell division. But if you have more than 40, I think, then you start showing symptoms of disease, because it basically makes a protein that won't fold properly. And so those neurons start dying. So it's also partly a mechanism of DNA replication as how these repetitive sequences can occur. So anyway, at least it varies as well from organism to organism, but it could be up to half or 75% or more of the genome is just made up of these repeats, which is kind of crazy when you think about it. I mean, and if you think about the cost of DNA replication and cell division, right? We're essentially making a whole bunch of DNA that we're not using. So why do we still have it, right? Why is it still there? I don't actually know. And the funny thing is, actually, kind of going even more off on a tangent, got a little bit of luxury today because we've got this and transposons, and they're both fairly short. Part of the reason why we may have that is because sometimes those repetitive sequences kind of hide particularly non-coding genes, which produce RNAs of different sorts, and they can be very, very short, which are very useful under stress, whatever that type is. Not stress like, oh shit, I've got an exam today stress, but more drought conditions or low oxygen, or some kind of environmentally induced biological stress. And you see that a lot in plants, actually. Some of the the plants that are best able to cope with, say, high salt levels or drought, have these previously unknown bits of DNA in them, which only become activated under certain conditions. So that's potentially a reason for that. Okay. Any questions so far? It's been a bit of a rambling kind of meander through some of this stuff. But really, I just want to give you the sense that things are a lot more complex and less easily understood than one would first think, right? And a lot more varied, right? Our genome is not a monolithic entity. It's extremely varied in terms of what it has in it and what it does, right? And how that can be compared from organism to organism too. And again, none of this is on the exam today, right? So this will be on exam 3. Okay, so essentially what do we have up for next? A little bit of genome organization stuff, right? So some words which are useful. You already know this because I told it to you already, but oops. Actually. And then we have 2 forms of chromatin. Got euchromatin. I always used to make the joke that you know, the EU was very open to new people and stuff, and then they shut their borders and they're not anymore. So, but that's the way I remember it. And then we have heterochromatin is tightly compacted. Transcriptionally. Right, so there's a few words which are important to remember as we're going through this. And so essentially, chromatin is the solution to the problem of the dilemma of eukaryotic DNA, right? And the dilemma is that eukaryote genomes are huge in terms of the physical space that they occupy, and the nucleus is small, right, in terms of the space in which that DNA can be stored. So we need to be able to store this DNA in, you know, really small space. But on the flip side, that DNA has to be accessible, because different regions of it are being transcribed at different times. You can't just pack it away and forget about it, because you actually need to use it. You also need access to it as well for replication during cell division too. So how do we solve that dilemma? And really, chromatin is the solution to that. And so chromatin is, basically looks like this, right? Don't worry about the bit at the bottom so much just yet. But chromatin is basically proteins. So we have these core histones. There are 4 pairs, 2A, 2B, and H4 and H3. There you go. While I remember. There's 8 of them in total. So 4 pairs of histone molecules, and I always think of them as meatballs, right? So they basically look like meatballs with some spaghetti wrapped around them, right? And they have, and this will, I think we'll get back to this later on in the course after the next exam, I think. From memory, it's hard to remember from one semester to the next. But these histones have their own properties. Actually, yeah, we definitely do talk about it. And those properties can be modified to control how tightly or loosely bound DNA is. So this is not just a mechanism for compacting DNA and storing it. It's also a mechanism for opening in terms of transcription and closing whole regions of the genome at once. My usual analogy, and I don't know how useful this is because I don't know how many people have seen this, but if you ever go to a library that's big enough to have a reference section, which is typically like the old books, like down in the basement or something. I think if you go to the Central Library in San Antonio, it's like this. Any really big university library tends to be too. But essentially, they're books that aren't accessed a lot, right? So they're in these shelves or rows which can move on wheels. They're really fun, actually, if you're like a real nerd, I guess. But you can basically have, I don't know, perhaps twice the number of books in the same space as if the rows were fixed. And so to get to one, all you do is you kind of push these rows kind of down the room to open up the one that you want to. You go get your book. Hopefully some of your mates aren't jerks and kind of shut it behind you. And then the next person along who wants to access a different book kind of pushes those rows back and then can get into that particular aisle to get that particular book. And the analogy between books and genes is a fairly decent one in terms of information, right? So we can, cells can actually modify the properties of these histones to do the same thing, right? To be able to open up a region of the genome to transcribe it, or to shut it down and stop it being expressed. And it can do that for big, big chunks of it. So it's a very kind of broad scale control of gene expression. One way of thinking about it is, for example, if you have a program of gene expression, say whatever results in limb development or eye development, and once those things are done, you don't need to express those genes anymore, right? Because you have 2 eyes, right? You don't need any more. Although I could actually do with another couple, to be honest. These ones are getting kind of old. But once they're done, they're done. So you don't need those genes. And so if those genes are grouped together more or less on a chromosome, which they often are, you can just shut down that whole region of the chromosome, never to be used again, right? It's done, right? That was, GenBio 1. We're not using that textbook again. Get rid of that. And then that also frees up space for the rest of the genome, which can be more loosely compacted, right? And so this is a very, very precise structure. It's always the same, but it's one that can be modified. It can be modified by moving these histone molecules around, like sliding them up and down like an abacus. It can be unwrapping that DNA, kind of looping it out so it's more accessible, right? But always it's going to be 8 histone molecules with another like a piece of cheese, essentially, histone H1, which is involved in that particular shape. And also the spacing of these apart as well, so they don't end up kind of beads right next to each other, but they're spaced evenly apart. And the reason why DNA wraps, actually, does anybody know why DNA wraps around histones and binds to it? Some people may remember from earlier courses. It's a pretty fundamental property of DNA. Can anybody remember what charge DNA has? It's going to dig deep in your brains. Negative, right. And that's actually what we use to do gel electrophoresis too. That's how that works, right? Because we apply a voltage difference and the DNA goes towards the positive end. As long as you got the right way around, of course. So histones actually have tails. They have little regions which kind of stick out like a kind of, there's little like balls, pom-poms, things like that with like little bits of fluff sticking out. Those bits of fluff are positively charged, and so that allows DNA to stick to those histones, right? So one of the main ways in which we modify chromatin structure is by fiddling with the charge of those histones, by either making them neutral or allowing them to be positive. There are other ways as well, but that's one of the ways. And we'll talk about that later on in another lecture. In terms of how much DNA, this one's useful. It's about 200 base pairs per nucleosome. So here's a useful bit of language. Nucleosome equals 200 base pairs of DNA plus so that's what a nucleosome is, right? And so essentially that consists of, or you could put it this way, nucleosome equals core particle, which is 145, typically 147, kind of varies, to 147 base pair of DNA. Not FNA, that's something else. And about to 60 base pairs of DNA, actually linker DNA. So that's another way of thinking about it. So essentially, as we saw here on this, on this particular image here, right, this would be a core particle. It's got the 8 histones which form that meatball. It's got 145, 147 base pairs of spaghetti wrapped around it and a histone H1, right? But chromatin isn't just core particles. There's also some DNA in between, the linker DNA, and that's what allows these core particles to be kind of wrapped around each other, right, to coil them more tightly. So again, remember, like from the beginning of one core particle to beginning of the next one is about 200 base pairs, give or take. But each core particle has about 145 to 147 base pairs. And that's, remember the DNA, actually, how wide is DNA? I think this is about 10 nanometers wide, give or take. I can't actually remember how wide DNA is itself. I know it's 10 nanometers long. No, 3.4 nanometers long for every 10 bases. But anyway, once you get these nucleosomes, you can start kind of wrapping them around each other, right? And this is really the form in which you would expect to see some of your chromatin, right? So this is going to be very much kind of in the process of being transcribed or replicated. This kind of beads on a string type structure, but typically, in general, this is what you would expect your chromatin to look like in any given stationary phase cell, right? It's going to look like this. Some of these are going to get pulled out to be transcribed. This may be even more tightly wound. As you can see here, right? So this is a more compacted form. And I couldn't tell you for definite how these two relate to chromatin versus heterochromatin, because it's actually quite a bit more complex than that. What I can tell you though is that at this point down here, when you have those classic X chromosomes, this is fully compacted, right? There is no gene expression going on whatsoever in this chromosome, right? That's why in cell division, all of the making of stuff that you need is done before M phase, right? So you can't actually make anything new after that because you can't get to the DNA until it's decondensed in telophase. Okay, so essentially it's kind of a dynamic process switching between typically these two forms, and maybe down to the beads on the string, but typically between these two in terms of compaction. And it very much does, even within a single nucleus, within a single chromosome, right? This changes back and forth fairly frequently, depending on needs and transcription. And also, this is also super cool. There are actually domains in the nucleus where each chromosome has its own particular spot in the nucleus, right? And so you can actually identify, this will be this blue one, for example, here is going to be one particular chromosome. This orange one over here is going to be another chromosome. And those chromosomes are actually stuck to the inside of the nuclear membrane, right? So it actually kind of retains them in place. They have a very much a higher order structure. And there are variations in terms of activity, right? So the bits of the chromosome closer to the center are more transcriptionally active. The ones to the periphery tends to be more less active. And then you've got all the structures, the holes and things that allow enzymes and the like to move and around in that, which I think is pretty cool to think about. And then finally, even in each chromosome, whether that chromosome is being used or not, we have variation along that chromosome in terms of its status, right? And this is a useful image. It's not a very good one because this is a chromosome in prophase, right? So if we were actually to draw a proper image, it would just be this huge kind of jangled mess, right, of spaghetti essentially, right? So that's why I use this picture, because it's easier to see, not that it's actually 100% accurate. But typically, the regions which are transcriptionally active are the ones on the arms, right? So if you remember back to linkage and mapping, right, there are more genes actually in the arms, right? So map distance is much more accurate in terms of how it relates to physical distance in the arms than it is in the centromere or the telomeres, right? Because there's many more genes, especially, obviously the telomere doesn't have genes, centromere does have, but they're very few and far between. And so the centromeres also and the telomeres are transcriptionally repressed. So they are heavily compacted into heterochromatin, whereas the arms are generally found in, pardon me, in terms of euchromatin. Obviously, that may vary, right? Because there's still this dynamic switching back and forth between the two, depending on need. But as a general rule, the arms are typically euchromatin. The core or the centromere is typically heterochromatin. The only exception to that is in ladies who have two X chromosomes. In every one of your cells, one of your X chromosomes is entirely repressed, is 100% heterochromatin. And that's to make sure that you don't have twice as much X chromosome gene expression as you should. Because males only have one X chromosome, and that's kind of like the level at which gene expression occurs from the X chromosome. So in females that have two X chromosomes, every single cell has one of those X chromosomes shut down. And it's random which one as well. So you're technically a mosaic, right? In terms of what alleles you're, not what genes your expression, but what alleles are being expressed in terms of phenotype from the X chromosome, which I think is pretty neat too. All right, anybody got questions? Questions? While we're going to swap between this and transposons next. If you do have questions that you think of later, feel free to bring them to class on Thursday. A lot of this is just kind of fact stuff. I just wanted to put some of this into the broader context because, I mean, this is biology, right? It's, you know, you can't really just break it down into little bits. It's all part of a bigger picture. Okay, so okay, hopefully you can all see the transposons PowerPoint now. We've got, what's that, 25 minutes left? Should be enough time to get through this. If not, we can go a little bit over. I don't want to go too far over because the exam will be available at 12:15. I don't want to cut people off. So, oops. Neat name for transposons are jumping genes. And so there's a couple of cool examples. One of actually was how transposons were discovered, which is in maize. So we've all done the corn genetics stuff where you look at, you know, colors of corn kernels and things like that. What you doing, dog? And what you'll see quite often is that those corn kernels are speckled, right? If you looked at any of those pictures for that lab that we did earlier on in the semester, and I said, you know, any hint of purple equals purple, right? And so the reason why, come up here. The reason why that is the case is that the speckling arises from the inactivation of a pigment gene, right? Via a transposon, TC1 I think it is, jumping into it, and it inactivates it, right? And that's why you end up with lack of pigment, but it can be in individual cells. That's why you end up with it not being uniform. Another good example is pigmentation in flowers. Couldn't tell you what kind of flowers because I'm not much of a flower person. But yeah, I remember what they look like. They're really pretty. Kind of like that sort of shape, you know, kind of cool patterns, colors. Typically, oh shit. Dog's licking my bed for some reason. Typically white and a darker color. So purple and white or red and white, things like that. They're very, very pretty. And they're called variegated flowers, right? So they have a different pattern each flower, essentially. And the reason for that is, again, the same thing. A transposon has jumped into one of those cells earlier on in flower development and inactivated that pigment. So all the daughters of that cell don't express pigment, right? So you can actually see if you go to, like, you know, fancy flower shop or whatever, and you see flowers that are not uniform in color, like, you know, red or blue or whatever, but they have cool and varied patterns, that's almost always due to transposons moving around and jumping into those pigment genes. Right. And so essentially transposons are little autonomous bits of DNA that can move around in a genome without any external help, potentially. It kind of varies sometimes from transposon to transposon, right? And so they typically, not always, but typically encode the means with which to move themselves around too. So they are actually pretty funky, and they're actually one of the causes for a lot of our, so-called junk DNA in our genome is essentially replicating transposons, just bouncing around. And back before CRISPR was discovered, it was actually one of the main tools. This is what I was working on way back in the day for making stable transgenic animals. They used enormous amounts in Drosophila, which is pretty neat. Okay. So transposons have, there are a couple of different types of transposons. There's DNA transposons, right, which, as you would expect, are made of DNA. In fact, at the end result, they're all made of DNA. And these can either transpose or jump by replicating themselves, so there'll be replicated transposons, or by cutting themselves out and sticking themselves somewhere else. That's non-replicative transposition. The other type are called retrotransposons, and there are literally thousands of different types of transposons of all shapes and sizes. But retrotransposons have an RNA intermediate, so they transcribe themselves into RNA, and then that gets copied back into DNA before that DNA gets inserted into the genome. And so there's some kind of typical characteristics of a transposon, right? So one is that they recognize a very specific DNA sequence. Some of those can have very, very short specific sequences. For example, is it piggyback? One of the ones that I worked on, AT. So anywhere there's an A and a T next to each other can be a site for transposition. And so they have this target repeat. That's what they will go into. They often have these terminal inverted repeats. Those are recognized by the third element, which is the transposase, the enzyme that actually transposes them. And that's often encoded by a gene in the middle here between those terminal inverted repeats. So again, there's a target sequence. When transposition occurred, that is essentially copied and creates two flanking direct repeats. Each transposon typically, doesn't always, but typically has these terminal inverted repeats. And if you actually read them, they are the copy of each other, right? And so this goes TGC-GAT, and this is TGC-GAT, like in the opposite direction. That's also a classic characteristic. So if you want to go find these, it's actually fairly simple to find them. You just look for these patterns. And typically have a enzyme or enzymes in the middle which facilitate that transposition, right? This is also one of the ways in which transposons end up getting stranded. And so if there's a mutation in that transposase gene, that transposon is now dead, right? In the absence of a transposase from another transposon related to it, it will not move, it will just stay there. So you can see how there are these relics essentially of activity in the past which just get copied with every generation and round of cell division. And so we just inherit them from our parents and their parents and their parents and so on. One of the neat things about this is that typically they make staggered cuts, right? And the reason for that is that these staggered cuts make these single-stranded ends, right? So essentially, the transposase will cut the DNA, creating a double-stranded break, but a staggered break. And then it will ligate the transposable element in to that break and then DNA polymerase fills in those gaps. And so when transposition has occurred, you end up repeating these repeats, right? These flanking direct repeats. Also of consequence is that not only if this is into a coding gene, not only when it jumps in, does it cause damage potentially in terms of mutation and gene function, but because these flanking direct repeats are copies, right? They're not there in the original sequence. Even if that transposon jumps back out again, it will leave a footprint, right? These flanking direct repeats will essentially be a little doublet, right? So for example, in piggyback, it goes from being AT to 80-80, right? And that footprint, because you've added bases, is still mutagenic, right? If it's in the middle of a coding region, for example. So not only when transposons jump in can they cause problems, and they frequently do, but also when they jump out as well. Anybody got any questions about this? This might be new to people, so I'm happy to take a little more time as necessary. This also got a researcher called Barbara McClintock a Nobel Prize too. So she was the first one to discover transposons, and she discovered them in maize. And the reason was that she could not explain kernel pigmentation by Mendelian genetics. And so when she kept doing these crosses, she kept getting weird results. And a lot of people just, blew her off. In fact, she spent a very long time convincing people that transposons were real. She did some really, really beautiful work. But eventually, people were like, oh, well, I guess you're right then. These things do exist. And as a result, she got a Nobel Prize. So that's one of those really cool Nobel Prizes that kind of rewards something that is truly game-changing in terms of discovery, right? And the idea, because this is what people struggled with, the idea that DNA was mobile, right? DNA is typically thought of as a static, repository of information, right? The only change occurs via mutation from external sources or DNA replication, right? Well, essentially transposons don't fit that dogma, right? Transposons are, in a sense, their own individuals, right? In a way, right? They have their own DNA and they kind of move around as they see fit. Kind of weird to think about it in that way because obviously they're not living, right? Their DNA, it's like saying viruses are living, but they do have independence, right? And they do replicate independently of, well, potentially of their host. So kind of weird to think about. Almost like DNA parasites in a sense, which is kind of weird. Okay, so this is DNA, or this would be a DNA transposon. Retrotransposons are a bit funkier, and typically these actually can contribute to a large amount of junk DNA because these can replicate quite effectively. And so they're retrotransposons because of this part, this, and I don't know if any of, actually, can anybody think of a disease that also has the word retro in it? And we're not talking about like wearing flares or something like that, but an actual disease. It's a class of diseases. There's one very famous one. Yay, retroviruses. Can anybody think of a retrovirus? Very famous one. Yeah, that's it, HIV. So HIV is a retrovirus, and it's an RNA virus which can copy its genome into DNA, and that DNA can get inserted into the host genome. That's why quite often HIV can go dormant, because, and that shingles, I think, is another one, actually. And then later emerge at times of stress or being immunocompromised or something like that. And so the cat fur, the same kind of yeah, that's right. Same deal there. And the reason is it's gone into your genome. Some DNA viruses do that too. It's not just retroviruses, but retroviruses do it via an RNA genome. And so for HIV, that's why we don't have a vaccine for it, because RNA replication is very error prone, and it produces a lot of mutations. And so the HIV virus changes a lot, whereas other DNA viruses and things don't typically, or not as much. So anyway, this retro part is down to this process called reverse transcription. And what that's all about is the transposon, or virus, if we're talking about retroviruses. We have a bunch of RNA via transcription, and then that RNA is serves as a template for a enzyme called reverse transcriptase, right? And that will copy that RNA into single-stranded DNA, right? We actually call that complementary DNA, or cDNA, in kind of molecular biology, and we use that a lot too. And then that single-stranded DNA gets copied and turned into double-stranded DNA. That's the same for retroviruses, and actually, in some ways, the mechanisms are similar. Retroviruses tend to insert particular sites in the host genome, and so once you have that double-stranded DNA copy of that transposon, the same process occurs. Staggered double-stranded break, filling in of the flanking direct repeats, and now you have a new copy of that transposon in the genome. So you can see not only can these things kind of jump around independently, but they can also replicate, create more copies, which then jump into different regions of the genome. And so the only thing which dictates where they go is way back here, this target sequence, which is unique to each transposon, or each type of transposon. So wherever you find that particular sequence, might be TAT or GGAA, for example, they're often palindromic, so they'll have a kind of read the same in opposite directions. Or yeah, palindromic will not be quite the right word. That transposon can insert itself, right? So you can see how you can get over time. Oh, it is right? Oh, cool. I thought it was. Sometimes I'm not entirely sure what, I think I know what words mean, but I'm not entirely sure if I'm explaining them the right way. So, and also in terms of molecular biology, what you can do, and this is what I worked on, is you can get a copy of that transposon, and you can actually take out that center part and replace it with something that you want to get inserted. Say a GFP reporter gene, right? That was one thing that I was after. Actually, what I was working on was a mechanism for sterilizing possums, believe it or not, because New Zealand has a very big problem with possums. They go around eating everything. Particularly like they're cute flightless birds, which didn't have predators until they came along. They eat their eggs, for example. So anyway, that's what I was working on. You can actually put cargo inside that, and as long as you can provide the enzyme, the transposase, separately by making it actually getting expressed in yeast or something, you can insert that modified transposon into the genome of your target organism, and it will be stable and inherited. Every single cell in that the offspring of every single offspring will have a copy of that that cargo, right? So that's what I worked on for a couple of years. I was in Australia actually. Didn't actually get it working for many reasons. It's not a trivial challenge. But until CRISPR-Cas came along, which is now the latest hotness in terms of transgenics, using transposons was the way in which people made transgenic animals, or one way, at least stable transgenic. And that's pretty much it from me. Oh, obviously, how could I forget? Not always, but often transposons can be mutagenic as well. And that's usually because they're big, right? So if you have a particular coding region or even a regulatory region at the beginning of a gene, which relies on proteins binding to that stretch of DNA in a particular way, having this big ass thing insert itself, and these can be several thousand base pairs in size. Some are even actually huger. These, you can get really, really long, big ones. Having this big chunk of DNA get slapped, inserted into a coding region or a regulatory region even, can just knock out that protein completely. And so, oh yeah, there's even that aspect too. Typically when they bounce around, if they do end up in or near genes, it results in a complete loss of function mutation of that gene, right? Which, depending on what gene it is, can have either piddling consequences, pigmentation, right, in flowers and plants, or pretty severe consequences. Also, one of the weird things is that because these particularly replicative transposons, they copy themselves. So they, oh, bloody cats are going off now. That transposon is going to be present in potentially different chromosomes, and it's the identical sequence. And so there is a risk, particularly for really large ones, that that will serve as sites of homologous recombination. And so they think that's one way in which we, over evolutionary time, we have chromosomes that grow or shrink in size, is potentially because of this erroneous homologous recombination during cell division, or during meiosis actually, which basically, as a result of that transposon being there, those chromosomes don't get lined up like so, they might get lined up like that. Or even you might get chromosome 1 getting lined up next to chromosome 2, which would obviously generate something completely different. So anyway, that's kind of a big broad brush overview of both genome structure and a lot of the repetitive DNA that's part of it, and also the role that transposons have in, I don't know, genome evolution, I guess, and a lot of that repetitive DNA. And that's pretty much it from me. Does anybody have any questions? No? Does anybody have any questions about stuff that's going to be on the exam for today? Because if you do, now would be the ideal time to ask them. All right. Okay, so that's pretty much it from me. I'm going to go and stretch my legs quickly, and then I will, the exam is due for, yes, it will end at 11:59 tonight, Tuesday the 7th. And yeah, thank you, Ali. It's PowerPoints 7 to 11 inclusive. Right, so not nothing from PowerPoint 6. Anyway, I'm going to end this Zoom meeting, and then I'm going to open up another one in about 10 minutes. And I'm just going to sit here checking emails and stuff, just in case anyone has any questions about the exam, which will open up in a couple of minutes. So if you go to your exams and quizzes folders, pardon me, hang on a second, Austin. In a couple of minutes, you'll see exam 2 appear and a PDF which you'll need to use to answer questions in that exam. Austin, yes, there is lab today. We will be starting the module series. We'll be doing module 1. It shouldn't take very long. It's really just going over the project in general and also a little bit of the specifics of it. So it shouldn't take too long, probably about an hour at most. But yeah, you will find that in the course content. Actually, what is the folder called? I can't remember off the top of my head. Let me just double check. Yeah, that's Lab 8 Module 1 is the folder where you'll find that. So have a quick look at that. And again, nothing crazy, but essentially introducing what we'll be working on for the rest of the semester. Cool. Does anybody have any other questions? No. Awesome. All right. So I'm just going to go get some more water. I'll be back here in about 10 minutes time, just hanging out if anyone has any questions or problems. And if not, I will see you at, let's say 1:45 for lab. Actually, let's say 2 o'clock, because I think that's what I've got in the meeting. All right. Let's stop this.